Welcome to MOVICShiny. Below, we will begin using this platform for multiomics analysis. First, we need to configure some basic information. Please enter the email address you used when applying and user account containing in confirmation email you received. This is our first login, choose Yes. If not, choose No. Fill in the cancer type and project name for the analysis. Here, we will demonstrate using the Bulletin BLCA dataset. Click the Create button to login, login successful. Click the Process button to prepare and process data. Click the Integrate button for data integration. Because the external validation or not parameter in the basic settings module is set to Yes, we need to proceed to prepare external validation data. Users can customize their selection based on their specific needs and choose the data types they require. Here, we choose mRNA, Yes, and upload manually based on our prepared validation dataset. Next, we upload integrated dataset, which can be downloaded from our GitHub. Click the Create button to create a folder for storing the external validation dataset. Click the Process button to initiate data preparation and processing. At this point, all data is prepared. Let's go to Get module to start multiomics analysis. First, the prepared data needs to undergo pre-processing. We process the TCGA and validation sets one by one. Next, we determine the optimal clustering number using the Get Clustering Number method. Users can choose to download the corresponding PDF images and R data result sets after completing. Subsequently, we perform consensus clustering using the appropriate technique. If consensus clustering is desired, we need to select two or more clustering methods. After configuring the parameters for the consensus clustering heat map, click the Process button to start creating the heat map. The next step involves evaluating the quality of the clustering results by calculating the sample-to-sample similarity of the obtained subtypes using Silhouette. Finally, we will generate multiomics heat maps based on the clustering results and pre-processed multiomics data. This concludes all analyses in the Get module. Next, click the Comp module button to perform comparative analysis of the obtained subtypes. Under Comp module, if you need to select the validation dataset for each step, it is necessary to first complete the analysis of Run Nearest Template Prediction or Run Partition around Medoids classifier of validation dataset under the Run module. Firstly, we will compare survival outcome between different subtypes, which requires the prior preparation of clinical survival data. Next, we will compare clinical features of each subtype on specified clinical variables, and clinical survival data should be prepared in advance for this analysis as well. Following that, we will compare mutational frequency of genes between subtypes, necessitating the preparation of binary somatic mutation data. The subsequent step involves comparing the differences in total mutation burden among subtypes, and again, binary somatic mutation data needs to be prepared beforehand. Additionally, we will compare fraction of the genome altered by copy number alterations in genes affected by copy number increase or decrease in each subtype, requiring prepared copy number alterations data. Moreover, we will use IC50 values to compare the response levels of each subtype to drug treatments in the GDSC database, and mRNA or lncRNA data needs to be prepared in advance for this analysis. Finally, we will assess the consistency between clustering results and traditional classification results, evaluating the quality of the clustering results, and clinical survival data should be prepared beforehand for this evaluation. At this point, all analyses in the comp module have been completed. Next, click on the Run module to proceed with downstream analyses for the clustered subtypes. Under Run module, the algorithm for differential expression analysis should be the same. Run a differential expression analysis among the clustered subtypes. Based on the results of the differential expression analysis, run a procedure to identify biomarkers, filtering for upregulated or downregulated biomarkers in each subtype. Perform GSEA to analyze the enrichment of gene sets, identifying upregulated or downregulated pathways in each subtype. Conduct GSBA to analyze gene set variation, calculating enrichment scores for each sample within each subtype on specified pathways. Use the model-free method NTP to predict the subtypes of samples in an external validation set based on the identified biomarker genes. After completing the subtype prediction on the external validation set, you also have the option to reselect TCGA and use NTP to predict the subtypes. Additionally, use another model-free method, PAM, to predict the subtypes of samples in the external validation set. Evaluate the consistency between the predicted subtypes and the clustered subtypes using similarity and reproducibility metrics like IGP. After completing subtype prediction on the external validation set, you can also choose to reselect TCGA and use PAM to predict subtypes. Finally, assess the consistency between the clustered subtypes and the predicted subtypes or between predictions using both NTP and PAM, utilizing the kappa statistic. Click the Finish button. You have completed the entire analysis workflow on the MOVICshiny platform. At this stage, you can download and organize the corresponding result files, including the RData result set, plot PDFs, and result tables, for further analysis based on the generated output. Additionally, if you have obtained NTP or PAM prediction results for an external validation dataset, you can return to the comp module to select validation for comparing subtypes in the external validation set. It's important to note that users should choose the appropriate analysis in the comp module based on the type of omics data available in the external validation dataset. Here, we demonstrate 4 types of analyses that can be performed with our validation dataset. If the analysis is not completed, you can follow the steps below to log in and again. When the program encounters an error, the platform will give the specific reason for the error, red error box, and a warning message, yellow warning box. Users need to hover over the yellow warning box, so that the 2 message boxes can remain on the web page without disappearing. If you still have questions about the use of the software, you can click the orange small question mark button of each parameter module in the upper right corner, or refer to User's Guide. So far, the use of the website has been all introduced. Now, let's try to perform multiomics analysis on specified cancers through this website, and we welcome you to point out errors or give valuable advice on our website. Thanks for your watching.